She may or may not jump on, and if she does, I'll have her introduce herself. So she's at the College of Western Idaho, and she's a microbiology person. And so we're trying to set her up so that next summer we always provide a fellowship to CWI. So the first year from Gut, it was Dusty, and last year it was Rebecca. And so I'm hoping that Elise will be our next sort of fellow that we support over the summer to do some microbiome research. So if you start to see her, that's where she comes from. But I say do we want to do a quick, just anything exciting or anything that anybody wants to share that they've done or that they're, if they want to share, a little round robin. If you got nothing to share, you can just go, you can just shake your head, go away. But any good things anybody wants to talk about?
My lightning talk presentation is over, so that's good.
Yay! Yeah, and you got some really good feedback. At some point, maybe our meeting today, we should look over what people's questions were, but great job. I heard good things about it. Sorry again that I missed it, but.
That's okay. I had over 70 people there, so I had plenty of people looking at me.
And thanks to the rest of the lab that helped provide some feedback and to Brianna, so it's great. Jim has recruited a computational student to help Deb. This is good news. I don't know where things are, but are you guys already making some plans? And
we have made contact. We are working on I am working on the scope of work, and I'll try to get some ideas from you in our meeting today. Okay. But yeah, we met. I'm super excited because I'm not the computational person for what she's really shown interest in doing with that part of the project. So very excited. Very thrilled.
Yeah, she's the kind of kid who like goes and does a whole bunch of stuff on their own. Like, so like yeah. you say here, can you do these things? And then like 20 more things get done. You go, oh, well, that, you know, that's nice. You know,
I felt like when I met with her, I was saying here, this is what I want to try to do. And I felt like she was thinking, well, that's really super easy. What else you got for me? Because she was kind of like, okay, we could just write a code. And I'm like, we could, but it would take me 20 years, so.
Yeah, that's awesome. That's exciting. Stephanie, you have a COVID lab happening?
Yeah, so I got offered a position as a lab manager to open up a clinical testing lab on campus. It'll address COVID shortages needs right now, and then hopefully later on, we're getting a license called a CLIA license, which will make it possible to do drug development and human clinical studies in the future for BSU. So kind of exciting. It should expand our research capabilities.
Nice.
And it'll be right across the hall from Jen's lab.
Right. Perfect.
Yeah, so I'll be going down to 10 hours a week for Jen. That project's been a little consuming right now. I know, I'm sorry. But hopefully this will all end soon and things will go back to normal. She doesn't, so.
There is no end in sight.
Yeah, well.
Okay, so anything else anybody wants to share? Jem, do you have anything in addition? I forced you to say something, so.
You know, my natural habitat is to like quietly be in the corner, right?
Hiding.
Yeah. No, actually, I'm working with Jessica in Eric's lab. She's got this data around this CPEB3 ribozyme that does like the self-cleaving, and they've got this like really cool visual representation of structure that you can identify because there's a change in fitness for different mutational pairings. And so you get this really cool picture, and it fits with this thing called a singular value decomposition, which is it's a imaging technique that they use to take really dense images and make them so that you can pull features out of them with low dimension, and so they're fast and it's kind of a cool technique. So I'm kind of looking forward to working with this group that I've got meeting every Wednesday to eventually see if there's a way we can apply some of these these matrix methods to reduce dimensionality of these really dense images and see if we can say, hey, I can predict a shape of an RNA because I've got some indication of the structure because I can see it in this picture. And so I probably know half of what I may, probably not even that, probably like 10% of what I need to know about it, but it's cool to be able to see this connection between, like, you've got this output data that came from real experiments, and you can like maybe apply some like mathematical technique to it and then computationally do it and do it really fast. And so I'm excited about that, but I'm geeky that way.
Forward to seeing what that looks like. Yeah. All right. Well, I'll let Jonas then just take over.
All right. Can y'all see the screen?
Yep.
Great. Okay, so thanks guys for coming. Today I'm going to be giving a presentation discussing the utilization of different diversity metrics to assess the composition and structure of microbiota. So this is obviously a huge, broad topic, and I'm not teaching an ecology class here, but I'm hoping that this presentation will serve as a good starting point to think about different diversity metrics and use it a bit as a learning tool, especially through the context of looking at microbial communities. So for the outline today, I'm going to be addressing diversity analyses as core components of ecological research, which you all probably know. Well, you all know, but sort of re-emphasizing why it's important to grasp these concepts and implement these concepts when you're performing various kinds of ecological research. Discuss how distinct diversity metrics are utilized. So one of the main goals I'm hoping to get to convey through this talk is to think about that different kinds of diversity metrics tell you different things, and there's no one metric that answers all of the experimental questions that you want to address. We're going to be using general examples to visualize microbiome diversity concepts. So I've created sort of a sample microbial community system that will be doing real basic, not even calculations, but real basic conceptual analyses on communities and see how that is represented by different diversity metrics. We'll discuss the utilities and shortcomings of diversity metrics, and if we have time at the end, interpret some real world data, so apply some of the concepts that we address. This presentation does run kind of long, but this isn't by no means, like a thesis presentation. I want this to be an educational tool, and we'll have spots in between talking about different metrics that I'd like to, if people have questions, we can discuss, or if things are confusing. So we'll do that. Objectives, so ideally be able to recognize the differences between alpha and beta diversity. You all probably know that, but it's good to reiterate these concepts. Grasp how diversity metrics are calculated. So huge disclaimer, I'm not a statistician, okay? I have applied diversity metrics to analyze different kinds of communities. And I think mostly the goal I'm trying to get across through this presentation is when you see output from metrics, so you have a computer program calculate these metrics, how do you interpret these metrics, and how can you use these metrics to provide information and answer different experimental questions? And yeah, understand how distinct metrics are evaluated to address experimental questions. Okay, so first and foremost, what is community diversity? Okay, in its broadest sense, community diversity is just variations in biological communities. So, we've heard time and time again, there are communities of plants, animals, microbes, metabolites, genes, I'm sure other kinds of communities. But basically, community diversity, just variations in these biological communities. And so diversity analyses enable qualitative and quantitative evaluations of individual communities. Okay, so analyzing what kinds of plants or animals or species or genes are present in that community and in what relative abundance is. And then also comparing these community properties across different communities or across samples. Okay, so how is community diversity analyzed? So we'll be discussing alpha diversity. So alpha diversity represents variations in diversity within a community. Specifically, we'll be looking at OTU rich or species richness, Simpson and Shannon diversity indices. And important to note that these diversity indices, there are many, many different kinds, but for the sake of this presentation, we'll be covering some commonly used metrics that you'll see a lot in the literature. Okay, beta diversity, variations in diversity between communities, and we'll be looking at Bray-Curtis dissimilarity and unweighted and weighted unifrac distance. And I'll repeat it multiple times throughout the presentation, but it's really important to understand alpha diversity represents diversity within a community. It's a set in stone value associated with one sample, with one community, whereas beta diversity is an entirely different data structure, provides entirely different information where it's a comparative metric between communities, and that'll make a little more sense later on. And through all of this, we're going to be thinking about these concepts through the lens of analyzing microbiomes. So microbiome are communities of microbes, bacteria, archaea, and fungi. We'll be focusing on bacteria. And for this presentation and for many microbiome analyses, researchers often evaluate alpha diversity first and then beta second, and we'll maybe touch on why that is in a bit. Any questions before we get started? That was mostly just review for most of y'all. Okay, so example framework. So this is the framework that I'm setting up the next slides where we'll be discussing the different metrics. This is entirely hypothetical, theoretical, oversimplified. By no means will the examples we're looking at moving forward encompass all the nuances of the different metrics, show you all the different kinds of relationships, but you got to start somewhere. So for the following examples, we'll be looking at 4 different samples or 4 different communities. Let's think about these as, hey, sage grouse. Okay, so we have 4 different sage grouse, alpha 1, alpha 2, alpha 3, alpha 4. And I am actually working on a project investigating microbial communities associated with sage grouse. Okay, so that's sort of setting up our example framework, looking at these microbiomes associated with a host. Okay, and for the following examples, we'll have bacterial communities composed of different color bacteria. And for the sake of this example, we're going to say that red, blue, green are closely related taxa, closely related bacterial species, whereas brown represents an outgroup. And you'll see why that's important here in a little bit. And before we get started as well, and I'll do this for beta diversity as well, it's important to understand the data format, how data is provided, how these diversity analyses are calculated from these data, and just sort of understand the underpinnings behind these values that are output. So here's a read count matrix. Rows represent individual samples, right? We have our alpha 1, 2, 3, 4. Columns represent, in this case, different sequences or different species of bacteria. You can think about it. So I completely made this matrix. Okay, I designed these communities so that various qualities of the metrics that we'll talk about are sort of emphasized based on these community structures. This is by no means representative of real communities. However, I think it'll be helpful. You can see the columns here represent different sequences. So our brown out group, we have that sequence, and then red, blue, and green are all offset by 1 nucleotide. So those are represent closely related sequences. When you're analyzing microbiomes, right, you may have next generation sequencing of 16S RNA gene amplicons, And one of the outputs when you get the data back from the sequencing facility and you do some processes, one of the first things you do is you build this read count matrix where you have all your different samples and you have the sequence counts or the read counts of all the different bacterial taxa.
Hey, Jonas, before you go on, I just want to like link to our chemistry people in here.
Yes.
So like the columns could also be particular compounds. And then in those cells, instead of raw read counts, this would be the area under the curve or the absorbance value or some other metric of a sort of count that goes in for a particular compound. So when you guys are making these align peaks, this is essentially the same table that you're making. I just want to like tie those two things together so that as you're thinking about these diversities, you're sort of swapping microbe for chemistry or gene or whatever it is.
Yeah, absolutely. And a lot of these concepts are applicable to other fields. I don't want to make it seem like the diversity metrics we're talking about here are universally regularly applied because I'm not super familiar with some of the other fields, but totally thinking about, you know, any type of community data where you have counts of individual traits across different samples, you know, thinking about these concepts, I think are very applicable. All right, let's see. Okay, so alpha diversity. So the first metric we'll discuss is richness. So you'll see this as species richness, OTU richness, whatever trait you're looking at richness, right? This is probably very familiar to everyone, but it's a qualitative metric. And it's literally defined as just the number of unique species or the number of unique traits in your community. So these circles represent our 4 different communities, K alpha 1 through alpha 4. We have our different colors here, and this is the visualization we'll be using moving forward. But this is literally the visualization of that read count matrix I showed you earlier. So just pointing out some real broad scale traits here before we get started. So our community 1, okay, we have our 4 different colors and even relative abundances. Alpha 2, community 2, we have the 4 colors, but with brown heavily abundant. Community 3, browns the entire community. Community 4, okay, we also have an uneven abundance structure, but with heavily abundance of green. And just to reiterate, red, blue, and green represent similarly related taxa. Brown represents the outgroup. Okay, so keeping that in mind, thinking about richness of these communities, okay, we're characterizing richness. Going down the line, our first community has a richness of 4 because they're all 4 taxa are represented in this community. Okay, moving forward, community 2 has a different abundance structure clearly relative to the first one, but the richness for that community is also 4 because all 4 colors are present. All 4 bacterial taxa are present. Okay, community 3 has a richness of 1 because there's only 1 bacterial taxa present. And similar to the first 2 communities, okay, community 4 here has a richness of 4 because all 4 bacterial taxa are present. Okay, so this is an ugly plot, but we'll talk about it. So alpha diversity measures, again, remember each measure is associated with each community. So one community has one alpha diversity value for whatever metric you're looking at. And so this is a kind of a icky box plot here, but just looking at this, okay, we have clearly 4 different communities with entirely different community structures. However, 3 of those communities have a richness of 4, and so they cluster together, okay? And then we have our 1 community that had a low richness represented by this dot down here. And this will provide some context when we look at the other diversity metrics for comparative purposes. So richness is probably super familiar, but does anyone have any questions? And richness is certainly used very often. However, you can clearly see it's not a super informative metric in terms of, you know, what's actually there, because our community 1, 2, and 4 all have the exact same richness. So if you only looked at richness while trying to identify your community, you wouldn't really know that much about that community. Okay, the next diversity metric we'll talk about is the Simpson Diversity Index, which we're specifically going to be looking at the Simpson's reciprocal index. And it's a bit confusing to me because when you look in the literature, Simpson Diversity Index and Simpson's reciprocal index are often used interchangeably. So I'll be referring to Simpson's reciprocal index as the Simpson diversity index moving forward. And that is also the way a lot of the literature presents it, but there are other versions of Simpson's index, which can be a bit confusing. But so the Simpson diversity index is a quantitative metric. I say accounts primarily for evenness in community structure, in the structure of community. That's not entirely true because richness also affects this measure. However, I think it's potentially easiest to think about evenness, how Simpson diversity relates to evenness. The values range from 0 to 1, 0 meaning low evenness, 1 meaning high evenness. And when we say evenness here, evenness purely means how the community members, how your species in the community are distributed. So a community with high evenness would have whatever bacterial taxa is present there in similar relative abundances, whereas a low evenness would indicate a high dominant structure, so few taxa in that community have a disproportionately high abundance relative to other taxa in that community. Okay, so we have our 4 communities here, and the epitome of your evenness, right, is this first community here where you have your 4 bacterial taxa, each in equivalent relative abundances. So for this example, community 1 would have a high Simpson diversity index. Okay, community 2, clearly it has a less even distribution of the bacterial taxa present. However, there still are 4 bacterial taxa present. Okay, there still is a rich community, relatively speaking. So this will have a lower Simpson index relative to the first one. So for this example, we'll call that a medium or an intermediate value. Community 3 is the definition of low evenness, right? It has a completely dominant community structure with brown being the only taxa present for this simplified example. So that would have a low Simpson diversity value. Okay, and similar to the second community, our 4th community here, even though it's composed of different relative abundances of the different taxa compared to the second community, it would also have a medium Simpson diversity index. Okay, because this value, the Simpson diversity value is purely based on the distribution of taxa present, not which taxa are present necessarily. Okay, so here's our box plot. The first box plot was hard to interpret, but here the way our box plots are shown, the horizontal line within the box represents the median value. The top and lower limits, the upper and lower limits of the box represent the quartiles around the mean. And then we have our outlier points outside of that. And so when you're characterizing alpha diversity, within a group, you'll often see box plot values or values represented by box plots. Any questions on Simpson diversity? I'd also like to point out, so the purpose of these exercises here, clearly we're not comparing across groups. We're not saying that certain communities that we're looking at belong to population 1 versus population 2. We'll talk about that later. This is purely just trying to emphasize, here's what the values represent, what they tell you about a community, and also important to note, right, each point here represents an individual community, and that value doesn't change. Okay, that value is associated with that community. Okay, and finally, Shannon diversity index. So similar to Simpson, it's a quantitative metric. It accounts for both evenness and richness. It's biased towards richness. So Simpson and Shannon are similar. However, we can think of Simpson, the prior metric we looked at, telling you more about the community distribution, right? The evenness, whereas Shannon holds a little more weight to the richness relative to the evenness. And let's think about that a little bit after we look here. So again, these examples, okay, thinking about evenness and richness, these examples are limited in the scope that we can talk about these diversity metrics, but here for our first community, okay, it would have a high Shannon diversity index due to it's very rich and also evenly distributed. Okay, community 2 here, right? We still have that high richness. There are 4 taxa present. There's a less even structure present. So that would have a medium Shannon diversity index. Community 3, okay, would have a low Shannon diversity index, right? It's not very diverse at all, highly dominant, only one taxa present. And then community 4 would also have a medium diversity index, so, or Shannon diversity index. So, okay, here's our box plot, right? This looks exactly the same as our Simpson box plot. Note how the y-axis does have different values. Simpson and Shannon are related, and I think this varies across fields that you're looking at. So when thinking about microbes, okay, if you had a low Simpson diversity index, okay, that means that you have a highly dominant structure, low evenness structure. You can think of bacteria as interacting with each other within that microbiome. So it would make sense, right, that if you had a highly dominant bacterial taxa within a community, it could be competitively excluding other bacterial taxa, hence lowering richness. Okay, so that's sort of why we're seeing this relationship in this particular example, how Simpson and Shannon, you know, are basically identical, okay, plot 2 and plot 3 here. But I really do want to emphasize that that is not always the case in microbiome studies, and also in, I'm sure, other fields, although I'm not as familiar. Okay, and sort of one of the final points I want to make looking at these plots, Kay, is that while Simpson and Shannon look very similar, richness here, our first plot, right, has a completely different spread of values, and actually doesn't have a spread of values for those 3 communities, right, because they have the same richness. So just looking at alpha diversity, right, depending on which metric, is actually telling you different things, and it's not sufficient solely to use one of those, just one metric, when trying to actually gauge the true overall diversity of the communities you're looking at. Okay, so thinking about the alpha diversity workflow, right, we went from our read count matrix, okay, so this is the same matrix we looked at before, and calculated the alpha diversity. So if you look at the 3 columns on the right here, Simpson, Shannon, richness, each one of those values is calculated based on your read count matrix, or your trait matrix, right? And each one is associated to an individual sample. Okay, so that's purely calculated from whatever data you have in that read count matrix. And to summarize, okay, so measures of alpha diversity reveal overall diversity of individual communities, right? I keep saying it. Each value is associated with one sample. What we were looking at here were estimates of evenness and richness, overall diversity. These measures enable within group statistics of pooled individuals. So what do I mean by that? We will touch on this later when we look at our real world example, but once you have your alpha diversity values for all your samples, right, you can then group those values based on a categorical variable. So you could think of, you know, different populations, right, population 1, population 2, and then you can calculate the mean, median, quartiles, variation of those values within groups, and perform statistics across groups, okay? I really want to emphasize that the box plots I just showed you is purely showing you, you know, what the values represent for an individual community and how those values can be visualized. Okay, you wouldn't be able to do stats on what I just showed you. However, using those, that fundamental process of calculating alpha diversity per sample, you can then start grouping and performing statistics depending on your system. Okay, and important to note, right, there was no information from these metrics on which specific taxa were present. And what I mean by that is that phylogeny was only used to define richness. Color did not matter, right? It didn't matter whether we had brown was the dominant community or green was the dominant community, okay? It was purely, you know, being able to distinguish between different traits to formulate those calculations to get those values. Okay, and again, each measurement is independent and singular. Diversity data for one sample, each alpha diversity related to one sample. Okay, any questions before we move on to beta? Okay, so we'll hop right into the beta diversity workflow. So it's fundamentally different than our alpha diversity data workflow. So first and foremost, we start with the same read count matrix, almost the same read count matrix. Okay, so this is a a count matrix of relative abundances instead of absolute count. So if you'll see, right, alpha 1 still has, you know, the 25% of each color in it. Alpha 3 still has completely brown in it. These were converted to relative abundances, which is commonly used, at least in microbiome studies. It's a normalization technique. Very conveniently, right, the communities that I designed here all had 8 bacterial taxa present, suggesting that there were the exact same number of sequence counts per taxa present. That's not going to be the case, right? And odds are each sample by sample will have variable read counts associated. So you won't have, you know, an accurate representation of how many counts exactly represent this, the precise abundance of taxa present. So converting to relative abundances is a way to normalize that in terms of analyzing community composition and structure. This is certainly a point of contention in the field, but commonly used, and so we'll be using that for our example framework. Okay, so, but important to note, right, this read count matrix is the same one that we used earlier, just converted to relative abundances. Okay, here are our 4 communities, nothing's changed. Alpha 1, Alpha 2, Alpha 3, Alpha 4, those are 4 different sage grouse. Okay, and rather than, instead of where alpha diversity, we calculated individual alpha diversity metrics per community. Okay, when we're calculating beta diversity, we perform pairwise comparisons to produce a distance matrix. So what does that mean? Basically, every community you're analyzing, so let's say we're starting with alpha 1, each community you're analyzing will be compared to each other community you're analyzing, and you'll calculate a value for whatever metric you're looking at. So what that means is that starting with alpha 1, okay, we'll compare alpha 1 to alpha 2 and get a value for whatever metric, depending on how you're calculating the metric, okay? Alpha 1 to alpha 3, alpha 1 to alpha 4, and similarly with all other samples, okay? So each sample has a single value for a given metric associated with its comparison with each other community being analyzed. So there is no one value assigned to a community because each value you get in your pairwise distance matrix naturally is associated specifically with another community. So fundamentally different data structure in your beta diversity metrics relative to your alpha diversity metrics. Okay, so transitioning to beta diversity, we'll look at Bray-Curtis dissimilarity. So this is a quantitative metric, accounts for specific taxa and relative abundances. Okay, values range from 0 to 1, a low value meaning communities share the same taxa at the same or similar abundances, right? A high value represents communities that don't share taxa and exhibit disparate abundance structures. Okay, and here's our distance matrix. And so I really want to emphasize, okay, where alpha diversity was pretty simple, we had 1 value per community. Beta diversity is more complex in terms of, at least for me, thinking about conceptually what these values mean. So I'm going to be showing you some ordinations for each metric. There is a ton of information you can gather from those ordinations that we'll talk about. I won't be covering all of them because, you know, there's tons of information, but I will be pointing out specific interaction or specific comparisons within this matrix that are comparable across the different metrics we'll be looking at and important for distinguishing between the metrics. So here's what our Bray-Curtis dissimilarity distance matrix looks like. Okay, and rather than box plots for alpha diversity, beta diversity, you often visualize your pairwise your distance matrix using an ordination method. So for the following examples, these are principal coordinates analysis ordination. Basically taking your values from your distance matrix that were calculated for each pairwise comparison and plotting them in 3 dimensions, or in this case, 2 dimensions, to visualize distances between the points. So the ordination is basically trying its best to transfer these distances from this pairwise distance matrix into a 2-dimensional plane that captures those actual distances when looking at cluster or the closeness or farness between points. So each point here represents one of our individual communities. And so just for this one, 2 main points I'm pointing out here. So the comparison between alpha 1 and alpha 2, keep note of the color here. Okay, so we have a value of 0.375. So without any point of reference, you know, that number doesn't really mean a whole lot. But when looking at 1 to 4, we also have a value of 0.375. So let's think about that, right? Our community, alpha 1, right, is our evenly distributed community. Its comparison to alpha 2, right? Alpha 2 has a lot more brown than alpha 1 and less of the other colors, and we get a set value. And then when you compare alpha 1 to alpha 4 has a lot of green relative to alpha 1. Okay, so it actually, alpha 2 and alpha 4 have the same abundance structures in the sense that one bacterial taxa takes up 5 of the 8 community members in that community. The colors are different, but when we're looking at Bray-Curtis dissimilarity, Bray-Curtis doesn't care about those colors, okay? It's purely comparing which taxa are present and relative abundance structures, hence alpha 1 to alpha 2 and alpha 1 to alpha 4 being the exact same distance. So taxonomic distance does not impact value. It didn't matter that brown was the outgroup, okay? Bray-Curtis is solely looking at distinguishing taxa and relative abundance structures. Okay, and again, there's a lot of information in this distance matrix. I'm only picking out two to really emphasize some of these concepts. Okay, any questions here? I think the next slide on weighted unifrac will provide a good counterpoint to Bray-Curtis. There are also lots of different ordination methods as well. And I'm not 100% sure on the finer points of, you know, the utilization of each, but I can certainly say principal coordinates analysis and NMDS ordination are frequently used, at least in microbiome studies, to visualize these metrics. Okay, next metric we're looking at, weighted unifrac distance. So similar to Bray-Curtis dissimilarity, it's a quantitative metric. It accounts for presence, absence, and relative abundance of specific taxa. And importantly, values are weighted for phylogenetic distance. Okay, so low values, again, similar taxonomic composition and similar relative abundance. High, different taxonomic composition and different relative abundance. Here are our communities, here's our distance matrix. And I think the difference in this measure is really, relative to Bray-Curtis, is really exemplified by the ordination here. So first and foremost, you can already tell it's a different ordination structure. And looking at the same comparisons that we were looking at in the Bray-Curtis dissimilarity matrix, so alpha 1 to alpha 2, we have a value of 0.426. Okay, so here's our comparison here. And so looking at the exact same comparisons that we identified in Bray-Curtis, okay, 1 to 4, we have a value of 0.329. Okay, so remember in Bray-Curtis, these two boxes had the exact same value. Here in weighted unifrac, we have different values, with 1 to 2 having a higher value than 1 to 4. So thinking about why that is, this measure takes into account phylogenetic distance, okay? And the distances are weighted for that phylogenetic distance, meaning that a difference between the difference between 1 and 2 has a highly different relative abundance between the brown, right? 1 has few brown, 2 has a lot of brown, okay? And we know that brown is the outgroup, okay? So that difference in relative abundance is really exacerbated in this metric. When looking at 1 to 4, okay, it's similar abundance structure in alpha 4, right? It's got a lot of green, a lot more green relative to alpha 1. But because of the similar relatedness between red, blue, and green, and our red, blue, and green here, right? The difference between the outgroup, brown, is less drastic, hence the lower weighted unifrac distance value here. So take home message here, right? The exact same comparisons between the communities, okay? But using a different beta diversity metric gives you different values depending on Bray-Curtis or weighted unifrac, even though they're both quantitative measures. Also importantly here, 2 to 3 has a very small, well, relatively small weighted unifrac value here because it does share similar relative abundances in that brown out group, which is weighted more heavily, right? It takes that into account. Okay, relative to 3 compared to 4, which is the highest difference or the highest distance, at least in this distance matrix, because there is such a disparity in the relative abundances of that brown out group, which again, weighted unifrac takes very seriously. So any questions with weighted unifrac, especially when compared to Bray-Curtis dissimilarity?
Yeah, so why would you choose Bray-Curtis over Unifrac ever? Because it seems like Unifrac has so much more information content to it.
It depends how much you value taxonomic relatedness in the traits that you're looking at. And, you know, I can't speak for other fields. I'm not sure you can do phylogenetic distance for communities of metabolites. Jen, maybe you can talk about that, but at least for bacteria, it depends whether you're purely trying to classify different community structures, or if you want to see whether communities share similar bacterial taxa. It depends how much emphasis you want to put on the relatedness of the taxa that you're looking at.
Yeah, and as you were talking, I was actually thinking, and I haven't looked for this specifically, but I don't know if it really exists for chemistry, but it seems to me that either 2 ways to sort of get at like relatedness, because it's kind of getting at a functional. You assume that maybe more closely related taxa might, I guess the assumption is that they have a shared sort of function, and so
which is a huge assumption that I'm not sure
right
is super.
But I think with chemicals, you that functionality is actually might be easier. So either compounds that share similar pathways and maybe have like similar functional groups, and or like using the Swiss ADME like approach of like how lipophilicity, what your lipophilicity is, and your like properties as the physiochemical properties, like those features might be an indicator. And I don't know how that would get included in here, but being able to think about how for the chemistry you can get at relatedness functionally to add to this type of analysis seems like a really, if people haven't done it, it's probably because most people don't think about mixtures in chemistry that much. But I think that could be something, Deb, that we think about connecting with your in silico approach to understanding the sort of potential function and pharmacology of these molecules, and then using that in some way to maybe calculate or do this type of analysis. And for Brianna, I think a similar thing, like you might find that there are certain structures that are much more similar to one another that, again, I think the thing that we need to sort out is how is it done for phylogenies? And then how do we apply that same information to chemicals? And I guess somebody needs to look into that.
Yeah, there are some programs though that do really similar analysis for metabolomics. They're all, I mean, anytime you're starting with a count table, you can do these types of ordination analysis, and you can overlay them on top of each other. And so it's not any different really.
So probably the first thing is like looking for the sort of taxonomic, like the nomenclature for chemicals that people, like I've seen these in the chemistry,
Yeah.
and that's probably the same, because then you have that like, how are they related, and you have that structure, and
Yep.
then that must just get applied the same way that the taxonomic groups are for microbes.
Yeah, and let me see if I can find, we did this. I did a workshop a while ago, maybe a couple of years now, with UC Davis, where we took count tables from different types of data, and we made cloud-based visuals that anyone can plug their data into. And I'll see if I can find that link, because it basically, any type of data that generates counting and a type, you can do the exact same visuals for each of them, whether it's a heat map or a PCOA or any of these different things. So I'm gonna try to throw it in the chat if I can find it really quick. But you guys can go to that website. They have had that workshop every year since then too, so they keep adding to it. But it's pretty neat because you don't have to learn how to code to use the tools. You can just go to the cloud, put in your own table and run it.
Well, this is great, Jonas. This is I like totally just like, oh, okay, now I get it.
Yeah,
So thanks.
this is really good, Jonas.
Awesome. Thank you. All right, so we have one more beta diversity metric we're looking at. Okay, so we just looked at weighted unifrac distance. So now we'll look at unweighted unifrac distance. So where Bray-Curtis and weighted unifrac were quantitative, unweighted unifrac is a qualitative measure, and it purely accounts for the presence, absence of specific taxa. Again, values weighted for phylogenetic distance, low values representing similar taxonomic composition, high different taxonomic composition. Okay, so here's our distance matrix. Okay, and you can already tell highly different values here relative to the other ones. And I think this is really exemplified by the ordination here. Okay, so relative abundance does not impact the value of the distance of the pairwise comparisons. Okay, you can see alpha 1 to alpha 2, alpha 1 to alpha 4, alpha 2 to alpha 4 all have a value of 0 in this instance because they share the exact same bacterial taxa. So it's kind of similar to richness in the sense that, you know, you're just counting colors. You don't care about relative abundance. What isn't shown here is that unweighted unifrac does take into account phylogenetic distance. So if we had other communities that were entirely composed of red or entirely composed of blue or entirely composed of green, we'd see different spreads of those clusters. But as you can see here, just using these communities, 1, 2, and 3 are completely on top of each other, with alpha 3 being over here on its lonesome, separating on this ordination. Okay, relative abundance does not impact value. So thinking about the utility of this metric, right? Maybe, you know, you care more about maybe the diversity of the community. Maybe you want an estimate of low abundance community members, and you put less weight on the relative abundances of community members. You just want to know if you know, a lot of the same traits are present in different communities. Unweighted unifrac provides a metric for that, putting a whole lot less weight for dominant community members and really just taking it at a qualitative level.
Wait, say that again for me. So like you would want it, like if you're trying to pick out the like outlier, the thing that's like that this would be a better approach maybe than the other one because it highlights the abundance issue, that you have this distinct group, but there's lots of them. That's why the alpha 3 is so distinct.
Right.
I understand that right? Okay.
So the way I think about this is unweighted unifrac gives you a good estimate of the overlap of species present in your communities without thinking about the prevalence of those species. So, you know, you could have a Bray-Curtis that clusters, all your points cluster really far apart because the communities are entirely structured differently, but you could have an unweighted unifrac that's tightly clustered, meaning that each community all had the same taxa present, but had highly disparate relative abundance structures. So, you know, this gives you a good proxy for whether species are present or absent across the communities you're comparing.
Yeah, so go back to your previous one. Can you go back to the other visual that you had? That one. Yeah, okay. So you would look at this and you'd be like, oh, I've got like each of my communities are like kind of a nice distance away from each other, and it wouldn't give you that sense of who overlaps as much, although those closer to each other are more similar taxonomically.
Yes, right. This
Okay.
is weighted for phylogenetic distance, okay? But right, you can't gain from this whether to the full extent of how much overlap in species present there are.
So you kind of need both to look at to like get, yeah, okay. I can see where
Sure.
both is helpful. Okay.
Right, and you know, what we just said there is applicable to these distances right here, right? Because these are different distances. 1 to 2 is a different distance than 1 to 4, even though the distances in the abundance structures are the same, right? Alpha 2 has 5 browns, alpha 4 has 5 greens. Their comparison with alpha 1, their distance value with alpha 1 has different distances because it is weighted for phylogeny. Bray-Curtis, remember, had the exact same value. Okay, so it's sort of each slice, each metric provides a different piece of evidence to trying to unravel the full complexity of a community. And so they're telling you different things. And, you know, as you said, right, if you just looked at Bray-Curtis or you just looked at weighted unifrac, you'd say, oh, these points are completely different. There's nothing similar about them. Maybe some are more similar than others, but when you pull in unweighted unifrac into the picture, you could actually be like, oh, 1, 2, and 4 actually share the exact same bacterial taxa. It just ends up that they're in different relative abundance structures.
Yeah, thanks.
Yep. Okay, so to summarize beta diversity, again, measures compare diversity, different aspects of diversity across communities, right? Each beta diversity metric was presented as a distance matrix because each community has a value calculated for a comparison with each other community in your data set. This enables intra and intergroup comparisons. So I've intentionally left stats out of this talk because I think that's beyond the scope of what I was trying to accomplish here. But do know, and we'll look at a real world example here on the next slide, there are ways to compare these metrics across clusters of communities, okay? We were just solely looking at the ordinations of our example communities, okay? And so one way to do this is to calculate the distance between group centroids. So if you had a bunch of different points on your ordination, you could group your points by different populations or groups or however you want to do it. And then you could statistically test whether those points overlapped or were distinct with the other groups. And so we'll look at a real world example. With alpha diversity, okay, you could estimate variation in your alpha diversity metrics by the coefficient of variation, so the spread of your points. With beta diversity, you can estimate variation looking at within group dispersion around the centroid, so we'll look at that in the next example. And no information on individual community diversity is provided by your beta diversity summary. Everything provided is through the context of a community's relatedness with other communities you're comparing. Not related, well, depending on whatever metric you're looking at, right? Also, your ordination will change as additional samples are added or subtracted, right? The ordination, the distance between the points, because each new sample you add will add to that distance matrix, right? Because it will be compared to all other communities in the data set. So your ordinations will look different as you add more samples. Compare that to alpha diversity, where each point on that box plot isn't going anywhere, right? It's staying the same because there's one value for each sample you have. Any questions on beta diversity? So the main point I want to get across, right, and you probably already know, is that alpha and beta diversity are two completely different entities with different data structures telling you different information. And then the metrics within those blanket alpha and beta diversity terms tell you very different things. And it's not sufficient to just look at richness, for instance, or just look at Bray-Curtis. All right, so last slide. So using the concepts that we just talked about, here's a real world example. So it doesn't super matter how this experiment was set up, but the main point here is that we have a blue control group and a red treatment group. So in this case, these are microbiomes associated with mosquitoes, either control being the blue, non-fungal, or red being fungal mosquitoes, meaning that I fed them these fungal spores. And so we're looking at how microbiomes change across this experimental treatment. You know, so we're separating our points now by a categorical variable. You know, this could be different populations of sage grouse you're comparing, different gut compartments, different gene families. I mean, whatever your experimental question is. But so here are alpha diversity box plots, okay? Simpson diversity on the left, Shannon diversity on the right. Okay, and what this is showing is that fungal exhibits lower overall diversity, right? The red boxes are significantly lower than the blue boxes. And taking what we learned about Simpson diversity, right? This plot on the left, a lower value means a less even community structure. So what this is indicating is that our red mosquitoes have potentially few bacterial taxa that are dominating the community. Okay, and that's also indicated by our Shannon diversity, right? Low Shannon diversity indicates low overall diversity, low richness in your system, whereas blue here seems to have blue here has high values for both these metrics, indicating high evenness in community structure and high diversity, or high richness and high overall diversity. Okay, so transitioning from our alpha diversity metrics to beta diversity. So this is a different type of ordination, but the concepts are still the same. We have again, our blue points, our blue mosquitoes, and our red points, the red mosquitoes. What this is showing is that red and blue are clustering separately. So blue isn't really clustering, but red is clearly clustering. And looking at the general locations of the colors, you can see that red and blue do separate out. So this is indicating disparate compositions and structures. I forgot to mention the left plot here is Bray-Curtis, the right plot here is weighted unifrac distance. Okay, so thinking about this, right, separate Bray-Curtis dissimilarity, separation of that ordination of our two groups here, right, is indicating that there are different bacterial taxa present and in different relative abundances. And then on the right, weighted unifrac distance is then taking into account that phylogenetic distance and saying, yes, there's different taxa present at different relative abundances, and the taxa that are present are phylogenetically distant between the two colors here. Okay, and taking this step further, okay, I touched on it on the previous slide, looking at within group variation for these beta diversity metrics, here used as a measure of centroid distance, quote unquote, distance to the centroid. And what that means is how far away is each point to its group center point. So you can think of, how far is a blue point relative to the centermost blue point. Okay, so you can see, blue's really spread out. The non-fungal is really spread out relative to red, which is really tightly clustered. Okay, so that gives you information regarding, firstly, blue and red are separated here, so different compositions and structures, and that red is super consistent in its composition and structure across samples within that group relative to blue that's all over the place. Okay, and so this is just using a box plot to represent those distances. Okay, so we're starting to get a picture of these communities. These are alpha and beta diversity analyses, right? And so taking it a step further, which I wasn't in the scope of the presentation, but taking it a step further, looking at relative abundance, right? So throughout this entire time, okay, we had our phylogeny. We knew that, some of the taxa were closely related. There was an outgroup. Some of the metrics took that into consideration, right? The unifrac distances, cared about phylogenetic distance, but none of this is telling you about what specific bacteria are we talking about, right? What families are we talking about, or what genera are we talking about? So the next step in at least microbiome analyses here is looking at the relative abundance of particular bacterial taxa. Real quick here, this bar plot on the left bar represents the non-fungal mosquitoes. So those are the blue mosquitoes, okay, the blue points. And then the right bar here represents the fungal mosquitoes, or the red points from the previous plots. Okay, and just looking from this, right, you can see completely different at the family level, completely different community structures, okay? And we can see here on the right, okay, this giant red proportion right here. And so the fungal communities are dominated by the bacterial family Burkholderia. Now, looking back at our alpha and beta diversity analyses, that makes sense, right? Because our Simpson was really low, indicating a low evenness, a high dominant structure. Our clustering for red was low, so indicating consistent communities across the fungal mosquitoes. And so what this is telling us is that all of our fungal mosquitoes were dominated by this one family, okay? So that in itself is interesting, especially when you look at the left bar here, right? Our non-fungal mosquitoes, we have all different colors, right? We have a diverse community structure here, Okay, and importantly, and I've sort of moved away from bar plots like this because they don't show you, variation in these families. But looking at our previous analyses, okay, our beta diversity plots, okay, blue, remember, was spread out all over the place. Red was super nicely clustered together, but blue is all over the place, high variation within group beta diversity. So that's implying that, yes, while these are the mean relative abundances and we have all these pretty colors, on a sample by sample basis, those relative abundances are highly variable. Okay, so we have two completely different types of microbiomes, right? In non-fungal mosquitoes, or in our control mosquitoes, we had a diverse community, but highly differential abundances of those bacterial taxa. Whereas in our fungal community, okay, we just had Burkholderia. It just took over the community. Okay, and naturally, you can take it down as far as many taxonomic levels you want to go. Here on the right here is genera within the family Burkholderia. So and this is by no means all the different types of analyses you can do with microbiomes, but certainly the workflow of going from alpha diversity, beta diversity, and relative abundance helps you paint this pretty complete picture of the types of microbial communities you're investigating. All right, that's the presentation. That's what I got.
Nice job, Jonas.
Thank you. Any questions or?
I didn't have a question. I had more of like a comment, especially the slide you're on right now. So you said when you first showed the first figures that it was significant, and I can see the stars indicating that significance. Do you have the p-values though, just to show just to have, because I know.
Yeah. So I intentionally didn't do any stats in this presentation, but so these were done since it was an experimental research project. Everything was in the lab, right? So these were this was a linear mixed effects model with random effects on the rearing container. So I had all these different replicate rearing containers with mosquitoes, pulled samples out of those rearing containers, and then did a mixed effects model accounting for that. So 2 stars means p less than 0.01, right? Beta diversity, I use Permanova, a nested Permanova accounting for the random effects of source containers. And that was, I think, super significant. It was some sort of significance. I think it was super significant. Back to the mixed effects model for these box plots here. And you don't often see these types, but I think it's important, to establish your variation in within group beta diversity variation. And yeah, this was just visualizing which families were there.
Okay, and then the other thing I had a comment on, which is super, super minor,
Yep.
is in the very beginning, you had mentioned the beta diversity being between group species. And then later you said it was among group species. So it's more just a little issue with verbatim and consistency in using the same verbatim. Just because for me, I was like, wait a minute. So that's just like a little super minor thing for me.
Yeah, I think the distinction that needs to be made is, right, a beta diversity value in itself is a value between two communities being compared. And so you have that value for each community comparison within your entire analysis. Now, once you actually start choosing your metrics, okay, those are actually looking for the bacterial taxa within those communities, okay, and depending on which metric you're using, right, that calculation will be different. But at the end of the day, the value that's produced from that analysis represents one pairwise comparison, and you end up having tons of beta diversity values, right? Yep.
Oh, yeah, otherwise it was very informative, very good.
Thank you.
Yeah. Any comments for you, Deb?
Oh, I didn't realize people left.
Yeah.
Yeah, we're about almost 10 minutes over. So.
Oh, sorry.
Oh, it was really good. I will have to watch the replay, but that's only because I was being distracted by the young one a little bit. So,
are we still recording right now, or?
We are actually, yes.
Okay. Cool.
Yep. Is there an option to be able to get your slides so I can post that onto the Trello board for the group?
I can email it to you.
That'd be great.
Awesome.
Cool. Well, I got to get to another meeting here soon too.
Awesome. Well, thanks so much.
Thanks, Jonas. I
Have a
appreciate
good
it.
one.
Bye.
See ya.